Sure. So let's get going. Can I ask everybody to turn off their video so everything can run as smooth as possible? Thank you. So welcome back, everyone.
Sorry, already. One of the speakers asked if maybe the other speakers and the co-hosts, if they don't mind leaving their cameras on. They said it feels better to see a few people actually moving. So if the other speakers don't mind doing that, that would be great.
Okay. My name is Fabian Berkey from Uppsala University. I'm one of the organizers of this first ESOP. So we're going to go straight back into it now with the last two talks for today. And first off is Susanna Porter from the University of California in Santa Barbara, and she will talk about some insights into eukaryogenesis from fossil records. So please, Susanna, you have 20 minutes, after which we'll take some questions. If you can share your screen.
All right.
Great.
Okay, does that look good?
Yeah.
Yeah. Okay, great. Well, I want to thank all of you for attending, and I especially want to thank the organizers for making this happen. I think this is a great idea, and I'm glad to hear that you're planning to continue with this even after the pandemic is over, whenever that may be. Anyway, the motivation for my talk is this question of eukaryogenesis. That is how the eukaryotic cell, with all of its the modern characters of the nucleus, the cytoskeleton, mitochondria, how it emerged, and whether the fossil record can have anything to say about this. Now, this has remained one of the biggest mysteries in the history of life, and in part this is because of the lack of known living intermediate taxa that would span the gap from the prokaryotic cell to the eukaryotic cell, and that would allow us to figure out the order in which all these different characters, I've listed some of them here, evolved. Now, I know we're in this sort of great age of microbial discovery, and we're already finding some fascinating new creatures that are giving us some insights into eukaryogenesis, but I suspect that even if we were to scour the globe and describe every living species on Earth, there would still be a problem with bridging this gap. And that is because extinction is pervasive in the fossil record. And a really good example of this is provided by the birds. If we were interested in understanding how bird morphology evolved, and all we had to go on was their closest living relatives, the crocodiles, we would be pretty screwed. We wouldn't have any idea how flight evolved or the order of all of these different characters. But because we have this fantastic fossil record of the dinosaurs, we're actually able to piece together the order of these different characters. So we know, for example, that feathers evolved long before powered flight, and the last common ancestor of dinosaurs, perhaps, or even earlier. And so just as these fossils are useful for reconstructing birdogenesis, we need fossils for reconstructing eukaryogenesis. The problem is, of course, that it seems almost like a hopeless task to use the fossil record to understand eukaryogenesis. For one thing, a lot of the characters that we're interested in have a really poor chance of getting preserved. But I think another problem is the way we view the early fossil record of eukaryotes. It's the prevailing view is that all of the action, all of the steps in producing the last common ancestor happened long before the first recognizable eukaryotes show up in the fossil record. Well, in this talk today, I want to inject a little bit of hope and suggest that maybe not all is lost, that there are some characters that do get preserved. I'll talk about a couple. And then for a lot of these characters, we can maybe use proxies for them, fossil proxies. And then I also want to argue that perhaps this prevailing view of early eukaryote evolution from the fossil record is wrong. And I want to present an alternative view that is maybe allow us to look at eukaryogenesis. So before I jump into that, I just wanted to quickly review some terms that I'm going to be using. I've been talking about modern eukaryotes. Instead, I'm going to use this term crown group eukaryotes. This is defined as the last common ancestor of all living eukaryotes and all of its descendants, including those that are extinct. Rest in peace. The last common ancestor of crown group is known as LECA, the last eukaryotic common ancestor. There are these extinct forms, the stem group eukaryotes, that branched off before LECA emerged, but after the split of the crown group with its closest living relatives. The stem group, crown group together, those groups are called total group eukaryotes. And then I also am going to use this term FECA, I don't know how to pronounce it, for the first eukaryotic common ancestor, and that's that first creature after the split with the closest living relatives. All right, so turning to this prevailing view, I just want to give you sort of a very broad strokes of eukaryote fossil record. The oldest widely accepted evidence for eukaryotes are microfossils that are tens of microns in diameter and ornamented that occur in rocks more than 1.6 billion years old, 1,600 million years. And then throughout this interval, the Mesoproterozoic, there are a number of fossil assemblages that have fossils that are interpreted as eukaryotes, but they tend to be low in diversity, low in abundance. And then beginning around 1 billion years ago, there are a number of fossils that are plausibly assigned to various crown group taxa. So for example, we have Bangiomorpha pubescens, which is shown here, interpreted to be a red alga. We have vase-shaped microfossils, a variety of species that are interpreted to represent amoebozoan, testate amoebae. We also see characters, innovations like mineralization and scales that I show here. And consistent with this sort of diversification of the crown group, we also see molecular fossils of eukaryotes, steranes. These are the geologically stable forms, hydrocarbons, of sterols, which are membrane lipids that are characteristic of eukaryotes. Okay, so here's this view. It's this idea of a delayed diversification of eukaryotes. That the assumption is that LECA, the crown group, appeared more than 1,600 million years ago, but the crown group, eukaryotes in general, remained rare and restricted. And when conditions changed, the environment changed, maybe oxygen increased, eukaryotes diversified in the Neoproterozoic. So here again is, I've just represented this delayed diversification early LECA model. Here you can see LECA appears quite early. And so all this action of eukaryogenesis happens back here before 1600. But I want to propose an alternative model that maybe LECA is late, that the crown group diversified, that basically LECA appears just before the crown group diversification, and that all of the eukaryotic fossils that we see back to 1,600 million years ago are stem group eukaryotes. So they may have had some of these eukaryote-specific characters, but they may not have. And you can see, if I map on this fossil record onto these different models, how important this model is for whether we can actually study eukaryogenesis. The early LECA model suggests that, all the action happened back here. There's still going to be stem groups around, but it's going to be a lot harder to piece together what happened through this interval when we have fossils. If LECA is late, then eukaryogenesis is happening through this interval. Okay, so how old is LECA? What is the evidence? If you read the paleontological literature, they're often citing ages of 1,600, 1,800 million years ago, but actually if you look at the variety of molecular clock analyses that have been done, the estimates span 1 billion years, and they're all over the place. There's an interesting story here, which is discussed by M.A. Et al. About the effects of Bangiomorpha, this fossil red alga, on the estimation of the date, and it pushes these dates a lot older. Interestingly, there's Bangiomorpha has been redated to a younger age, and there's suggestions that it shouldn't be interpreted so specifically as a Bangiophyte, and when you make up for both of those discrepancies, actually the discrepancies disappear. So perhaps LECA could be as young as, say, 1,100 million years. There's another problem with this early LECA model and a long sort of restricted interval of crown group occurrence, and that is that phylogenetic models that are basically sort of simply looking at the probability of crown group and stem groups persisting suggest that for a clade to survive to the present day, so by definition being a crown group, it needs to get off to a flying start. It needs to basically appear, rapidly diversify, and that's the only way it's going to persist. Conversely, it's very unlikely to have a crown group originate and then persist for hundreds of millions of years at low diversity. It's just too likely to go extinct. This is some great work by Graham Budd and Richard Mann, and I highly recommend these papers. Okay, so let's, for the sake of argument, assume that LECA did appear late, say 1100 million years ago, and ask let's look at these proxy records and see when we can say different characters evolved. So I'm going to look at 4 different proxies, different characters: cysts, a complex cytoskeleton, sterile synthesis, and aerobic respiration, which is a proxy for mitochondria. So starting with cysts, the resting stage is common in many eukaryotic life cycles, and this figure here by Schopp and Schilde show that cysts are widespread in all the major eukaryotic supergroups, suggesting that maybe LECA formed cysts, or there was some underlying capacity in LECA such that cysts evolved over and over again within the crown group. We can identify cysts fairly easily in the fossil record by the presence of medial splits. You can see this here, basically equatorial split dividing the fossil into 2 halves. We also see these well-developed circles, pilomes in the wall of the fossil. So I looked at representative fossil assemblages from 1650 million years to, up to 720 million years ago, and look for the presence of medial splits and a pilome. And you can see that basically medial splits and go all the way back. Pilome almost goes all the way back to basically the start of my data set. So that suggests that that cysts, or the capacity to evolve cysts, was present at least 1600 million years ago. What about a cytoskeleton? Well, one proxy for a cytoskeleton is evidence for the cell being able to actively change its shape. And I consulted with Alistair Simpson and Andrew Roger, who helped me a little bit talking about this, what would be a good proxy for this. And one suggestion was, well, we know in the modern protists that the formation of spines, for example, in these diatoms, it happens by extension of sort of cell-like finger-like extensions of the cell, where they form the spines and then withdraw the cell membrane. So this is evidence of active shape change. This has been suggested by Javot et al. As evidence for a cytoskeleton when we see these fossils with these various spines coming off of them. Okay, so spines is potentially a proxy for the presence of a cytoskeleton. Now, I should mention that this question of a cytoskeleton has become a lot more interesting with the discovery of genes that were thought to be specific to the eukaryotic cytoskeleton in some Asgard archaea. And this report by Amachi et al. Showing branching processes coming off this Asgard archaean. And so it's hard to sort of know where to draw the line between a eukaryotic cytoskeleton and what would be a fossil proxy for that versus a character that occurs maybe before Theca. So I'll be agnostic about that and just see what the fossil record shows. And not surprisingly, if we record the presence of just spines in these different fossil assemblages, we see that they go all the way back again to the oldest eukaryotic fossils. Again, this is maybe not a surprise. There's a little bit of circularity here in that the way we are able to recognize these early eukaryotes is in part by identifying these specific characters like spines that we think are evidence of eukaryotes. But anyway, we can say this character goes all the way back here, more than 1600 million years ago. Okay, turning to the sterines. So these circles show the record of the presence of sterines in these red circles up here. And then these empty, these hollow circles here are studies where the authors have recovered biomarkers, hopanes, for example, that are made by bacteria, but no detectable sterines. This pattern has been generally interpreted in support of this delayed diversification model. The assumption is, well, we know LECA was already around. It's making sterols. Sterols are plausibly thought to be present in the last common ancestor of eukaryotes. So sterol synthesis was occurring. We see these fossil eukaryotes. And so the fact that we're not recovering any sterines from these rocks indicates that eukaryotes were really low in abundance. And here I just grabbed a quote from a paper looking at sediments from 1100 million year old, assumed to be a paleo lake. And the quote is, "It remains a puzzle why these eukaryotic denizens preserved in the fossil record did not leave an appreciable sterine record, and why, despite seemingly favorable environmental conditions, eukaryotic productivity was so low that sterine-hopane ratios have been found to be 0. You can see that assumption there. Gee, this is a great place for eukaryotes to live. We see the fossils, but surprisingly, there's no sterines. Well, I want to suggest an alternative, and that is maybe there's no sterines because eukaryotic sterol synthesis had not yet evolved, and that we can use this record, in fact, to constrain in time both maximum and minimum age when eukaryotic sterol synthesis occurred sometime in this interval shown here. Okay, finally, mitochondria. Well, I think it's easy to argue that a proxy for the presence of mitochondria is evidence for aerobic respiration. I'm not arguing the converse, that if you don't see evidence of aerobic respiration, it means mitochondria hadn't been acquired. But I do think if you see evidence for eukaryotes as aerobic, it suggests mitochondria is around. So what's the best proxy for aerobic respiration? Well, it's evidence that eukaryotes lived in oxygenated environments. And luckily we do have proxies, geochemical proxies for local redox conditions in the water column. So this is developed by Simon Poulton and Don Canfield. If you take a sample of mudstone or shale and you look at ratio of various iron mineral species, that this is an empirical evidence suggests that it's below a certain ratio, the whole water column was oxic. Above a certain ratio, may have been oxic at the top, but anoxic below. Okay, so this is pretty simple. If you see fossils in a sample that has oxic water column, suggests that those eukaryotes were living in an oxygenated environment and therefore aerobic. It's not totally clear cut because both the fossil assemblage and the geochemical proxy are integrated through time. So you could imagine a situation where there's brief anoxic conditions, flourishing of maybe anaerobic eukaryotes. But in general, I'd say if you see evidence of this over and over again in a fossil in a unit, suggests there probably were oxic aerobic eukaryotes. I should note that the converse is not so easy to interpret. If we have anoxic samples with fossils, it's not clear which of these situations is going on. It's typically assumed to be that middle case, but it could be the other possibilities as well. Okay, so just to test if this is going to work, I have data from the Chuar group in the Grand Canyon about 780 million years ago. So there should be aerobic eukaryotes around. And sure enough, there is evidence for diverse and abundant eukaryotes in oxic as well as anoxic environments. Also, this is work by my student Tina Woltz. There's concern about, well, was there preservational bias? Would we not see fossils as often in oxic conditions? But it turns out that the preservation is just as good in these oxic versus anoxic samples. Okay, so looking at the record then, there's not a lot of studies that have combined the presence of fossils and the redox conditions of the shales, but there are a few. It's tend to assume that you can use the eukaryotic fossils themselves as a proxy for oxic conditions, which I disagree with. This is work by Hedda Agich, who's a postdoc in my lab. She's been looking at the Bilat supergroup in Canada and finds diverse eukaryotes in both oxic and anoxic environments at this time. So that suggests that we do have aerobic eukaryotes. Going further back in time to Mauritania, it's a little more hard to interpret. Most of the samples suggested largely anoxic water column. We see abundant diverse eukaryotes, relatively diverse. This time there's a couple of oxic samples where there looks like there's some eukaryote species. Going further back in time, 1400 million years ago, there's a unit from Russia, entirely oxic samples, with a couple of fossils that maybe could be eukaryotes. But notably, we know there's a much greater diversity of eukaryotes. They're not around this time, they're not present in these samples. And then going even further back to more than 1600 million years ago, again, this is data by Hedda, where she finds that the fossils that we know for sure, we're pretty confident are eukaryotes, are only found in the anoxic samples. And then if we look at the oxic samples, there are these smooth-walled, simple forms known as leiospheroids, which could be possibly just prokaryotes. Okay, so there's a lot of possible explanations for this. Could be sampling bias, counts diluted by prokaryotes. Could be that these oxic environments are a bad place to live. But I want to suggest that we should at least consider the possibility that maybe back during this time, 1600 up to even 1100 million years ago, maybe eukaryotes had not yet evolved aerobic respiration. Maybe they hadn't yet acquired mitochondria. So I know I'm out of time. This is just sort of summarizing the records of these four different four different characters, map them onto a tree. In a lot of cases, we can only place minimum age constraints that suggest that cytoskeleton are at least 1600 million years old. Mitochondria, aerobic respiration could have evolved as late as 1100. Sterols maybe appeared about 1100 million years ago. And I'll stop here and just sort of the final takeaway being that I don't want you to lose hope in fossils being useful for this question. Thank you.
All right, thanks a lot, Susanna. Very interesting. Please raise your hand if you have questions. Just going to give a couple minutes to people to raise their hand. All right. So we're going to start with Daniel Mills. Unmute. Daniel? I guess Daniel has disappeared. And we're going to take the next one then. Anna. Anna, can you ask a question?
Okay. Hello?
Hi.
Hello.
Susanna, thank you for the great talk. Very interesting. I'm curious about your model of the late LICA. I think it's possible, and I think it has been done, to trace separately these features of modern eukaryotes, such as mitochondria, sterols, cysts, some kind of sterile pathway, cysts, cytoskeleton, and maybe indeed molecular data will also show like different timing. What do you know about that? What do you think?
I see. So you're saying use sort of molecular clocks to actually date timing of appearance of some of these different characters.
Exactly.
Yeah.
Because I know that and you mentioned that a lot of studies were focused on the molecular timing of the appearance of LICA, like a whole thing. Yeah, but what about if we break it into several characters?
Yes. Great. So the only case that I'm aware of, and I'd love to hear if anyone else knows of studies, but there was this interesting study by David Gold et al. Where they tried to date the timing of sterile biosynthesis using molecular clock. And I think you can see here on the screen, they estimated, interestingly, the presence of simple sterile biosynthesis back to like 2.4 billion years, you can see here, which is around the same time as this great oxygenation event, big rise in oxygen in the atmosphere. Of course, there's wide age constraints on this. And then the other thing, talking to David, this is this is dating the appearance of specific genes that are involved in the sterol synthesis pathway. And I think there's some evidence, unpublished, that this pathway was present in the earlier than 800 billion years ago, consistent with this. But it doesn't provide a constraint on the evolution of the entire pathway. So anyway, I recommend that you check out this paper. It's the only one I'm aware of that sort of looks at this question. But again, there's sort of, the devil's in the details, where they're dating the timing of specific genes involved in the pathway, and not when the pathway itself was assembled. Does that make sense?
Thanks. Next question is from Jürgen. Jürgen, I think you can ask your question now.
Yeah, thanks, Susanna, for the great talk. I really enjoyed it. So my question is kind of related to the previous one. You used many different factors to date the origin of LECA. And I was wondering whether you could comment a little bit more on the steroids. I mean, they are also used for, I guess, for the origin of metazoa, but like for sponges. But it's known that there are bacteria who synthesize the same steroids which live in symbiosis with the sponges. So there's a high risk to mix it up.
Yeah.
What is your opinion on that, to avoid mixing it up?
Well, that's right. So I didn't mention this, but sterols are known to be synthesized by some bacteria. But looking into that, it seems that the general opinion is that those those sterol synthesis genes were acquired by lateral gene transfer from a eukaryote. That eukaryotes, that bacteria picked this up from the eukaryotes rather than the other way around.
Oh, really?
Yeah.
Okay.
Okay. So let's move on then. All right, we have one question from Tom Cavalli-Smith. Tom, you can speak now, I think.
Hello, Susanna. I'm really glad that you have now changed to accept that probably LECA is much later than many paleontologists have thought in the past, as I've argued for nearly 2 decades.
Yeah.
But I think that the evidence from spines and cell projections for the cytoskeleton that you cited is not very convincing to me. And I really think we could just basically put stem eukaryotes no earlier than about 1 billion years as well, which greatly simplifies things. And I don't want to give all my reasons for that, but I refer you to a hugely long paper that is now out in Protoplasma, which discusses fossil records and a lot of the molecular claims for dates earlier than that, and also suggests how eukaryotes may have evolved.
Yeah, so I mean, I struggled with this question of what is a good proxy for a cytoskeleton, and I think I probably emailed Andrew Roger and Alistair Simpson more than, well, they were very gracious, but answering my questions. I mean, I basically sort of, I realize it's an open question, what is a good proxy for a cytoskeleton, and it may be that we'll make some discoveries in prokaryotes to suggest that yes, indeed, that these spines, these branching spines that have been used to suggest that these fossils are eukaryotes actually were present before Feka, and therefore are not, these fossils, that would, as you suggest, that would take away one of the supporting pillars for interpreting these as part of the total group eukaryotes. Again, also with the cysts thing. I mean, as I pointed out earlier, it's sort of this interesting question of, and I made a point of talking about not when the first eukaryotes appear in the fossil record, but the first recognizable eukaryotes. So it's a question of when do these organisms, when do total group eukaryotes get to a point where they're making structures that are recognizably eukaryotic, right? So eukaryotes may be in the fossil record. It could be Feka is very old, and eukaryotes are present in the fossil record 2 billion years ago. We just can't recognize them because they're not showing characters that are distinctively eukaryotic. As you say, the alternative is possible, that the characters that we see in these fossils that are 1,600 million years old, the spines, the cysts, that we think, oh, these are eukaryotic characters. Well, maybe we'll find that they're not actually specific crown group characters, that they go even further back in time, and then we'll have to reassess, where we think total group eukaryotes actually does show up in the fossil record.
About 1 billion years or later, perhaps.
Yeah, maybe. I'm presenting sort of, what I think is sort of what is maybe widely accepted right now, that cysts and these branching spines, these large ornamented fossils are specific to total group eukaryotes, but maybe you all will prove me wrong with your new discoveries.
Thank you.
All right. Thanks, Tom. So we have a question from Puri Lopez Garcia. I think you can ask your question now, Puri.
I can unmute. Okay. Can you hear me well now? So
yeah.
Thank you for an interesting talk, and I have actually two little questions. One is that you assume that mitochondria are require oxygen, so are aerobic, but you have many mitochondria that, in addition to carrying out aerobic respiration, do other type of energy-getting reactions. So you could still have mitochondria without the need of using aerobic respiration on one hand, but also you could have micro-oxygenasis, even if you don't record a net oxic environment. And still, if you have, let's say, some type of early eukaryotes being aerobic, close to oxygen producers, say cyanobacteria, you wouldn't detect them in the fossil record. So that's my question. And second, a comment and a question on the sterol side. So how confident are you that sterons really record eukaryotes and not bacteria? So you mentioned that true, some bacteria have sterol synthesis genes, but they acquired them from eukaryotes. And actually, that's not quite so, because there's a paper early this year showing that, because we have many more bacterial genomes, these genes are present in other bacterial phyla. So it appears that sterol biosynthesis is broader in bacteria than we used to think. And that may mean something for interpreting sterane. What can you comment on that?
Yep. Great, thank you. Well, thank you for bringing up that mitochondria aerobic respiration thing, because I did try to be careful about that. What I want to suggest is that if you find evidence for aerobic respiration, that is definitely, I think, in eukaryotes, definitely an indicator, I hope, correct me if I'm wrong, that there's mitochondria present, right? So you can go one way, that if you see evidence for eukaryotic fossils in an oxygen environment, you can assume aerobic respiration, then we can assume they have mitochondria. But we can't assume the opposite. So, so the fact that, so in this figure here, the fact that if we don't have, well, I don't think we have definitive evidence of eukaryotes living in oxygenated environments through, say, a lot of the Mesoproterozoic, it doesn't mean that mitochondria hadn't been acquired yet. But what we can say, though, is by 1 billion years ago, this is from Hedda Agich's work, we have eukaryotes, it looks like, living in oxygen environments. We know that the mitochondria must have appeared by that time. So that's all I'm saying. It's permissible. It could be any time before that. We only have hard evidence like that, which is common in the fossil record. With the sterols and bacteria, this is something where I'm just trying to keep up on the literature and figure out what the sort of prevailing view is. And my sense, reading a lot of these papers, is that the sterol synthesis pathway is thought to have been assembled in the ancestor to eukaryotes, so it was present in LECA, or maybe there was some amount of lateral gene transfer early on, but the phylogeny suggests that it was pretty far back, and that maybe some of these genes have been passed around to bacteria, but and some of them are making sterols. But yeah, if it's, this is sort of a big discussion in the biomarker community about, well, if we have sterols, does it really mean that it's eukaryotes?
There's a recent paper by the group of Damien DeVos, who saw many of these genes in diverse bacterial phyla. So this means this makes it more unlikely that these genes come from eukaryotes and not the other way around.
Come from prokaryotes.
Okay, so the idea that they have is that because these genes are more widespread than we thought in bacteria, eukaryotes got them from bacteria, not the other way around.
Yeah. I mean, it's an interesting, it's an interesting puzzle then, why they do show up so late in the fossil record. In a way, I might flip it around and say, well, that maybe the evidence suggests that that sterol synthesis originated in eukaryotes. Since
you might say also that these are groups of bacteria that are maybe not that abundant, and so that they see that much in the fossil record.
Yeah.
Until they just bloom in eukaryotes. Thank you very much.
Thank you, Puri.
Okay, so we have a few more questions here, but we're running over time, so I'm gonna allow 2 more questions, but ask for brief questions and brief answers if that's possible. So Mark, it's your turn.
Okay, thanks. Really nice talk. Nice to see a good overview. Just a quick question. I thought sterols needed oxygen for production. There was an exception paper, not obscure, but a chemistry paper that you can make it anaerobically. So generally, I thought then, so the thinking is maybe also then need mitochondria if there are sterols being present. So your 2 lines barely overlap on the sterol and the mitochondria one. So any thoughts?
I think those lines barely overlapping, that is, for the mitochondria, that's sort of the permissible latest appearance of mitochondria is about 1 billion years old. And so if mitochondria evolved any time before then is what I'm arguing. So it makes sense that mitochondria would have appeared and aerobic respiration would have appeared before the appearance of sterols. I guess I'm not too bothered by the fact that they don't overlap. I would be bothered if there was some maximum age constraint telling us mitochondria had to evolve after, 800 million years ago, and sterols were present before. Let me see. The interesting thing is that a lot of these open circles here, where you don't see sterols, some of these are environments where we know we have oxygenated water. So we know the environmental. We know that there were eukaryotes around. They were living. There was oxygenated environments around. There's no sterines.
Okay, so last question from Andrew Roger. Andrew, only if your question is short. You can ask your question. Yeah.
Great talk, Susanna. Just quick question about the Protistology paper that just came out saying 1 billion-year-old Protistology in Nature Ecology Evolution, end of February. So that's 1 billion years, and you got a multicellular chlorophyte. How does that impact on all of this?
Yeah, I think that it's consistent with the Bangiomorpha pubescens, the red alga. So that's been redated to 1050 million years ago, 1.05 billion years ago. If we have total group red algae at that time, we should expect to find total group green algae at that time. I don't think everyone necessarily buys that that's definitely a crown group green alga. But it's, I think it's consistent with the whole story that we'd expect to see green algae. And LECA, it's possible, is not too much older than that. I mean, you could still have LECA, say 1100 million years ago, green algae, total group green algae, red algae at about 1 billion. That was work by Ching Tong. It was great, really good paper.
Thanks.
Okay, thanks everyone, and thanks a lot, Susanna. We had one more question from Alexis Smirnov. Sorry, Alexei, I don't think we're going to be able to take your question at this point, so I'm going to lower your hand.
Chat.
Susanna, can I ask you to unshare your screen so we can move on to the last talk for today? Great. So next on the line and our last speaker for today is Varsha Mathur, a PhD student in Patrick Killing's lab at the University of British Columbia. Varsha is going to talk about plastid evolution in deep branching Apicomplexan. So please, Varsha, stage is for you. You have about 10 minutes.
Is my screen sharing okay? Is it working well?
Yes, it works for me at least.
Thank you. Well, thanks everyone for staying around for the last talk of today. As Fabian said, my name is Varsha and I'm a PhD student at Patrick Killing's lab at the University of British Columbia. And today I'm going to be talking about new insights into the origins of apicomplexans and plastid evolution. So kind of taking it back to Ross's talk, we're back at apicomplexans, and as most of you probably know, they are a large group of parasites. They're obligate animal parasites, and they cause some pretty devastating diseases, the most famous of which is malaria. And recently, they were even shown to be core symbionts of corals. So they're very specialized parasites, and they have these specialized cell structures and compartments. You can see here a diagram on the left from Ross's paper. And they have all these really specialized compartments, such as the apical complex, and some endosymbiotically derived organelles, such as the apicoplast and the mitochondria. Now, the apicoplast is particularly interesting because it informs us of the evolutionary history of this group. It is a remnant non-photosynthetic plastid, so it tells us that this group of parasites evolved from free-living photosynthetic ancestors into this group of parasites that we see today. And for a long time, it was really hard to make inferences of how this change took place, until about a decade ago with the discovery of Chromarobilia and Vitrella brassicoformis, shown here in the light micrographs. And they're the closest photosynthetic free-living relatives of the apicomplexans. And they really allowed us to make inferences about what genes were lost as parasitism evolved and what the photosynthetic ancestor of the apicomplexans might have looked like. And so that's kind of where the field was at when I started my PhD 4 years ago. And although we had a bunch of nice genome sequencing data from the more medically important apicomplexans, such as Plasmodium or Toxoplasma, Cryptosporidium, and now we had new genome data from Chimera and Vitrella, we still had a whole bunch of missing data from deep branching taxa. Particularly, these are those that infect invertebrates, such as the gregarines. And so this was actually the main goal of my project, to sample in these deep branching lineages at the base of the apicomplexans, as well as some of these groups here that have a sort of unknown taxonomic placement. And so when we actually have samples from these kinds of groups, we can reconstruct the origin of parasitism, as well as how certain characters evolved, such as the plastid. So in order to do so, we used single-cell transcriptomics. As I said before, these are very understudied parasites, so they're not in culture. So we actually had to collect the cells from the animal host itself. And this was by far the most fun part of my PhD. I got to travel to some really beautiful places, go scuba diving, collect all sorts of invertebrates from polychaetes to tunicates to sea cucumbers, cut them open, and pick the gregarines. And so the gregarines actually have these really large feeding stages in the guts, so you can pick them up with a microcapillary pipette. And so we usually do that in the field, take pictures of them, and then bring the samples back to the lab, carry out cDNA synthesis, do a whole bunch of quality control where we have to make sure that we're not getting too much animal contamination. As I mentioned before, these are gut parasites. They're really bathed in animal juices. And then we prepare libraries and we sequenced on the Illumina MySeq platform. On the left here are some of the micrographs of some of the single cells that we sequenced, and this came out in our paper last year. And as you can see, they have quite a diverse morphology even between them. And then after this, we do a whole bunch of bioinformatics, assemble these transcriptomes, predict proteins, and then we search for 263 eukaryotic-wide gene markers, manually curate every single gene tree, removing paralogs and contaminants, concatenate all this data, and then build a phylogenomics tree. And so this was a tree that we got. We placed these 6 new taxa into an evolutionary context, but very quickly after our paper came out, this was already old news. Jan's group here, Jan Januszkovic, also around the same time published a whole bunch of really nice Apicomplexan transcriptomes from Gregorines and some other deep branching taxa. And so the obvious next step was to combine our 2 data sets together to make this really, really beautiful Apicomplexan framework. So this is the tree that I'm presenting here. This is a maximum likelihood phylogeny of the Apicomplexans and their relatives based on 195 nuclear protein markers. All the species shown in bold are the new species that were added. The ones in red are from our current biology paper, as well as some unpublished transcriptomes, and the ones in blue are the ones sequenced by Jan's group. And as you can see, we've really, really improved sampling of the Apicomplexans, especially these deep branching groups now. We have a new clade here that is basal to the coccidia and the hematozoa, which have a lot of the medically important Apicomplexans. And this group includes Rhytidocystis, Margociella, and Aggregata, Merocystis, and these specifically are parasites of marine invertebrates. Another big thing was our sampling of the Gregorines. We now have a whole bunch of transcriptomes from the Gregorines, which were once really elusive, all the way from the eugregorines as well as the archigregorines. So now we can really say unequivocally that the eugregorines and the archigregorines are indeed a monophyletic grouping. However, we still are uncertain about the placement of Cryptosporidium. Maybe even more samplings needed and some topology testing to sort of finalize where Cryptosporidium would fall within this. But by far the most exciting discovery from both our papers was that some of these parasites that were previously described in the literature as either Gregorines or other unknown groups of Apicomplexans are not Apicomplexans at all. So Symbion X here is sister to the free-living Chromera, and Vitrella and Pyridium is sister to the free-living Vitrella. We also have a new clade here that is basal to all the Apicomplexans and the Crompedelids, called the squirmids, which was termed by Tom Cavalier-Smith. And here they are, this includes the Digyellum and Platyprodium. So these are just another independent origin of parasitism. So over the past year, we've kind of gone from a scenario like this, where we considered there to be a single origin of this parasite, to a sort of polyphyletic Apicomplexan-like parasitism. We also have new taxa added in, such as Nephromyces, that was sequenced by Claudio's group, and this new marine invertebrate clade, and we have a whole bunch of taxa added in here. So now we know that Apicomplexan-like parasites have evolved at least 4 times independently, and we see a massive, massive convergent evolution in morphology. These squirmid parasites, as well as Pyridium, they look just like any other Apicomplexan in their morphology and the way they infect the animal, in their life cycles. And so there's some sort of maybe predilection in this group for this kind of parasitic form. And so the next question we wanted to ask is, sure, now we know that a lot of these parasites look like each other, but do they also have convergence at a molecular level? And to do so, we looked at plastid evolution across this group. Because Apicomplexan plastids are one of the hallmark features of this group. They're very well studied. It's a secondary red plastid that's massively reduced in gene content. They've lost all photosynthetic ability, but they still retain the plastid and a small genome. The reason they retain the plastid is because the parasites depend on it for the biosynthesis of these 4 metabolites, isoprenoids, type 2 fatty acids, iron sulfur clusters, as well as partially heme synthesis. So the next step was to take all this new transcriptomic data and search comprehensively for the components of these 4 pathways. So here we have a figure adapted from our current biology paper. Up here are those 4 pathways. Each slice of the pie is a component of that pathway. And just to be clear, sometimes they're sort of missing, but that's because this is transcriptomic data, so missing doesn't mean it's not there. But overall, we see some really interesting trends. First and foremost, we were able to answer a big question, which is whether Gregorines retain a plastid at all, because it was shown in Cryptosporidium and Gregorina that they definitively have lost a plastid. However, both in Jan's paper and our paper, we were able to see that one of the clades of Gregorines, that's the marine Gregorines, such as Lecadina, Pterospora, Lancasteria, do retain a type 2 fatty acid from the plastid. And the archigregorines that include Selenidium and Sidlachia retain all 4 of these pathways. At the same time, we do see multiple losses of plastids across the tree, so at least 3 losses within the Gregorines. And when we look at the Apicomplexan-like parasites, they too have a similar plastid dependency on these 4 pathways. If you can see here, Digiolum has all 4 pathways, so does Peridium. Platyprodium has lost the type 2 fatty acid pathway but retains the rest, and Symbion X only retains the isoprenoid pathway. So we see some patterns of loss and retention, but at the same time there is a similar dependency on these 4 pathways. And so the next question was to say, what about the genomes? What about the plastid genomes? Do they retain the similar kind of plastid genomes as other apicomplexans? Have they reduced in the same way? So we sequenced the plastid genome of Peridium. And as you can see here, it looks exactly like an apicoplast genome. It's converged to have the same exact sort of gene content, architecture, structure, and it even uses a non-canonical genetic code where UGA encodes tryptophan, as we see in coccidian plastids. So just a huge amount of convergent evolution. But at the same time, I'd like to bring up some interesting exceptions that we found as we sequenced some more of these deep branching taxa. And one such exception was in Margociella. So Margociella is part of this marine invertebrate clade that's basal to the coccidians and the hematozoa. And Margociella has an extremely unique apicoplast because it's the smallest one we've ever seen. It has an extremely high AT richness, the highest ever seen as well. And what's really surprising about this plastid is that it's lost all 4 plastid-encoded RNA polymerases. And this has never been seen before. In fact, it's been proposed that the RNA polymerases being in the plastid genome is one of the reasons that it's retained at all. At the same time, we also found an interesting exception in the corallicolids, the coral symbionts, where they retain chlorophyll biosynthesis genes in their plastid genome, but no other components of photosynthesis. And so, as we explore more and more of this deep branching taxa, the patterns of gene retention and loss in plastid is actually turning out to be more complex than previously thought, and we might have to reevaluate how we think apicomplexan reductive evolution actually takes place. And with that, I'd just like to thank all my co-authors at the Keeling Lab, Martin Kalisko, Leander Lab, and Mark, Arnie Camino, and Kevin, who helped us out with the sampling. Thank you.
All right, thanks a lot, Varsha. Great talk. Do we have any questions? If yes, please raise your hand. All right, question from Jan Januskovic for you.
Hey Varsha, it was a very nice talk. Well, I I just wanted to correct one thing. Actually, my paper, I wouldn't speak of my group. My dear collaborators, mostly from Russia, of course, are very much independent on their own, and I also want to say some of them have actually pushed this field quite a bit forward in recent years. So check out their papers that I think complement what you've talked about a lot on the ultrastructural level, as well as looking at the biodiversity. So my one question would be about this plastid genomes. First of all, I wasn't sure you commented on the extreme AT content in the end. So if you could say more about it, specifically about this Margociella, and also why do you think these genomes are still there?
Yeah, thanks for pointing out the papers by your Russian collaborators. Yeah, they're really, really beautiful. And they do a lot of TEM and microscopy, sort of following up on these weird Apicomplexan-like groups. In terms of the AT richness of Margosiella, I know that you, in your paper as well, you sequenced Rhytidocystis. And in the plastid context that you found there as well, they were really highly AT rich. So I think this is quite a fast evolving plastid. Maybe there's a relaxed selection on it. And in terms of why it's being kept, there still are essential protein coding genes like ClpC, SUFB, and TUF-A that are still retained on it that might be preventing its loss.
All right. Any more question? I don't see a raised hand here, but I have one or one question at least. I think that you mentioned that you now think with your new phylogeny that parasitism evolved several times independently in the group. Is that what you said? So why do you favor this explanation versus a couple of reversal to free living?
That's a good question, because now the tree does kind of look like there's more parasites than free living.
Yeah,
I think I would argue it's a harder transition, right? These parasites are really, really reduced in their metabolism, and like they're really reliant on their hosts. So to go back to free living, they'd have to acquire a lot of genes by HGT or some other mechanism. So I think it's a much more of a complex shift evolutionarily.
But that would be an interesting one.
It would be.
Yeah, cool. So, all right. Question by Chris Lane.
Hey, how's it going? Any evidence that they've any of these groups that are now or non-parasitic have any bacterial endosomes?
The non-parasitic groups, sorry?
Yeah, sorry. The ones that, so in follow-up on Fabian's question about the potential reversal of parasitism, is there any chance that they've got some bacterial symbionts?
In Chimera Vitrella, for example?
No, not in Chimera Vitrella, in some of the Gregorines that you were showing that.
Right. Well, in our data so far, there's been no evidence of that. We haven't seen any bacterial sequences that would indicate that.
But it's all transcriptomic?
Yeah.
Okay, so you're doing a poly A pull down?
Exactly.
Okay, so you may not see.
Yeah, exactly. Might not.
Okay, thanks.
Okay, so we're gonna have two more questions for today. Final two questions. First question is by Sonia Rickert.
Hey Varsha, thank you for that nice talk. I wanted to know, have you had a look now with the different clays that you have there, and how, if anything, what you found so far, does it mean anything for the ecology of the Gregorines, or where you find them, or the other organisms, in where they are, intestine, where the coelom, or what they do in the host?
I think that's the next big question, right? Like, I think with our data right now, we can't say too much, but we need to get more microscopy and more cell biology to actually answer those questions, especially for the sort of those squirmid groups, right? Platyproteum and Undigyalum. Yeah. Is their parasitism the same as the Gregorines, even though they superficially might look like them? These are still open questions.
Thanks.
All right, so final question for today will be a question by Adrian Reyes Prieto. I think you can ask your question now.
Okay, well, nice talk. My question is very simple. It's about the plastid genome of Margoliciella.
Mm-hmm.
So do you find any evidence that the RPO genes, or the RNA polymerase genes, are now in the nuclear genome and your transcriptomic data?
That was the first question, right? Like, how are they transcribing this apicoplast? Have the genes been, because in dinoflagellates, that's what's happened, right? The RNA polymerase genes are now in the nucleus and then targeted back. But we found no evidence of that in our transcriptomes. But then again, these are transcriptomes, it's not complete genomes. Another possibility was that they had acquired some other kind of RNA polymerase. And so we searched for all sorts of RNA polymerases through our data. Again, didn't really find anything. And so a third option might even be some sort of dual targeting of the mitochondrial one. And that's been seen before in land plants. So that's another option. But yeah, we do need more data to back this up and figure out what's going on.
Okay, thank you so much.
Okay, thanks everyone. Thanks again, Varsha. So that's it for today. So on behalf of the organizers, including Patrick Killing and Javi de Campo here, I wanted to give a big shout out to all the speakers today. We really did an awesome job. I think it was an enormous success. I also wanted to thank everyone for tuning in and participating today, questions and everything. So this was only the first day. Tomorrow there's a continuation to this. So we hope to see you all tomorrow, same time for the second act and final day of this online event with even more exciting protist research. So see you tomorrow and goodbye for now.
The recording has stopped.